molecule polynucleotide DNA structure, right? So what I'm showing you on this slide, to screen share. Okay, you see my slide, right? Everyone sees my slide?
Yeah,
yes, sir.
Yeah, so DNA structure. So if you see here, so we have two base pairs. So double-stranded DNA has base pairs. Okay, so G is bonded to C and A is bonded to T. So the important thing here is that the bonding is hydrogen bonded. And if you see GC pair, so it has three hydrogen bonds. And adenine and thymine have two hydrogen bonds. Okay, and also if you see these structures very closely, so these hydrogen bonds are linear in the sense that the donor and acceptor and the bridging hydrogen, and all the hydrogen bonds is linear, means that this is actually most stable hydrogen bond in any living cell. So all of these hydrogen bonds, all three GC and both hydrogen bonds in AT are really strong. Okay. So, but the fact that GC has more hydrogen bonds than AT, okay. So GC structures are more stable than AT. So this is an important fact. So if you have GC rich, okay, rich DNA, so we have higher melting, okay, higher stability, okay, and typically the stability is measured in terms of what is the temperature required to separate these strands, DNA strands. In double-stranded DNA, you have two strands anti-parallel hydrogen bonded to each other. So these strands, if you increase the temperature in solution, so they get separated. So the temperature at which these strands get separated is called the melting. And the process is the melting, and the temperature at which these strands get separated is called melting temperature. So if you have a for a given size of DNA, if you have GC rich, then it has higher stability and higher melting temperature. Okay, melting temperature. This is an important aspect typically experimentalists keep in mind who work with DNA sequences in terms of either in vitro chemical reactions or let's say processes involved in GC-rich sequences inside the cell. So any enzymes, okay, which need to separate these strands in vivo, okay, for example, for translation of DNA into RNA or polymerization of DNA, okay. So where actually these strands need to get separated, so these enzymes exert basically need to put more energy if you have to break GC bond rather than AT. Okay, this is an important aspect. So if coronary is AT rich, means so relatively less stable. Okay. So this is only in comparison with GC. Okay, otherwise DNA strands themselves are really stable because that is okay. So today's class is again coming back to this structure. So if you see this structure, so polynucleotides have base, sugar ring, and phosphates. Okay, connected to bases are connected to sugars, and sugars are connected to phosphates, and phosphates actually connect to nucleotides. So, but if you see the structure very closely, so there are a lot of single bonds. Okay, what I'm seeing in the slide is if you see in the phosphate connected to sugar, there are two bonds. Okay, so three bonds actually, single bonds. So along which rotation can occur very easily. And the base is attached to the sugar, so again two single bond. So the rotation can really occur at that single bond. And then sugar again in turn attached to phosphate to to the next nucleotide. Again, you have two bonds, two single bonds, which have to be used. So total, there are about 7 single bonds around which rotation can occur, okay, in these molecules. So which gives rise to, again, flexibility. Okay. So even within double-stranded DNA, though there
Sir, excuse me, in the bond number 1, won't there be any resonance or double bond character in the phosphate part?
No. So what I'm showing is P double bond O. LO is phosphate, O is oxygen.
Yeah,
Okay,
yeah.
this bond is a single bond. Okay, so you're writing scribbling something. Who is scribbling? Okay, on the slide. Okay, that's fine. But anyway, so this is single bond. Phosphate, oxygen is single bond. But there is other phosphates, which are not really bonded. Okay, so you have, I think, one with O minus, right? Okay, and maybe another is double bond. But the backbone, let's say called backbone, where actually the connectivity is going from phosphate to sugar to phosphate. So these bonds are actually single bonds. Okay, so despite double-stranded DNA has 2 strands, and the connectivity is between the strands is through the hydrogen bonding, which restricts them to form, let's say, certain structures, okay, where actually the rotation around some of the bonds is restricted. But nevertheless, they form actually rich structures. Okay, of course, well known is Watson and Crick double-strand structure. So in addition to these single bonds, the picture at the bottom shows that how exactly the base connected to the 5-member sugar, okay, where the base is connected to a single bond, again rotation can occur. Okay, so even in this, you see the bases can be in relatively 2 stable conformations. So one is called syn, another is anti, in which you see that adenosine is connected is in split, basically 180 between cis and anti. But more or less in purines, okay, pyrimidines. So it's mostly the anti-conformation is stable. So now if you add this complexity to the double-stranded structure which we learned through earlier class, which is called Watson-Crick base pairing. Okay, Watson-Crick base pair. Okay, so which is actually more stable kind of base pairs, GC and AC, because hydrogen bond is perfect. But so now if you allow okay, relaxations through these single bonds, so there are other structures which are actually possible. So Watson-Crick paired DNA, base pair DNA, okay, is very well known as BDNA. Okay. B, I believe, basically. Okay, so B form. Okay. But what are the other forms of double-stranded DNA are possible? Are shown here is one is A form and another Z form. Okay, so the B form are the parameters which we know. So it's basically 2 periodicity, right? So one is 3.4 angstrom distance between bases. Okay, so stacking distance between bases. Base, let's say this called base distance between 2 bases along the chain. So other periodicity is 34 angstroms. Okay, so where actually the 10 bases actually complete one term. And again, this is right-handed basis. So this is again called helical DNA. This is right-hand. So this is a more stable DNA, but nevertheless, the other forms shown here, A form and Z form, are also actually occur. So A form can occur in less aqueous or dehydrated. Okay, so if you remove basically water, okay, so either you have less water content or you add substances to solution where exactly water content is less. So then there are small changes in double-stranded BDNA, okay, leads to ADNA. So what does it do? Again, it is same right-handed helix, double helix. Okay, double helix. But however, the spacings, so in first case, base to base is 3.4 angstroms, and complete 10 bases almost complete one turn, okay, along the helix axis. These two parameters are actually different. So the table is also given on the right side. So if you see, it's basically it's 3 1 okay. So this is 24 angstroms, or this corresponding to roughly 10.5 bases. So 10.5 bases complete one turn, but in the A case it's about 11. No, it's 11. Correct. So 11 bases. Okay, so number of is very close. But however, so this helix is bulge, means that the diameter is increased. So at the same time, it can be compressed. So what is shown here is 3 structures, A form, B form, and Z form for 13 nucleotides, if I remember. 36 base pairs. So if you take 36 base pairs, so B form has certain, let's say, length along Z axis. A form, because its diameter is larger, so it's basically compressed. The same number of base pairs now gives rise to less length along the axis. So that's about 26 angstroms. Yeah, no, diameter is large, 26 angstroms. Compared to the B form, diameter is 20 angstroms. So the number of base pairs which complete one complete turn is 11 bases. Okay.
So, sir, I have one question, sir. These A, B, Z forms are forming at different water concentration.
Essentially, A is at B is physiological conditions. Okay, A is less either less water or dehydrated. So typically, it occurs in crystal structures where, let's say, you have to crystallize this DNA. So then crystallization means what? So you have more molecules packed together with less and less water. Yes.
Okay, sir. So only concentration is not the factor. There are other factors also.
Concentration of what you mean?
Water.
Yeah, so water content. Water content is less. Basically, how do you reduce water content? Either you increase other substances so that the water content is less, or you pack these molecules tightly so that water per, let's say, DNA molecule is less. Okay. So if you see, another interesting fact is that in B form we have what are called major and minor grooves, 2 grooves, right? You clearly can see in this picture. So major groove is basically larger space and which is actually kind of deeper, and minor groove is basically kind of less deep. But in the A form, okay, because now it's compressed more, so the major groove even becomes deeper and minor grooves become shallow, almost like disappear. You can see minor groove almost is not there compared to the B, you know. So the blue is actually phosphate backbones. Okay, yellow atoms, you don't see those are phosphates. Okay, phosphor atoms. But blue is basically kind of phosphate. You can really see that minor group clearly in B, but A you don't see. But zDNA, which is completely opposite. Okay, so to A and B, in the sense that this is left-handed double helix. Okay, all the double helices, left-handed double helix. Okay, so you might wonder that why, how can you have the 2 strands? You can understand B to A transition very easily because if you compress the helix, let's say from top to bottom, so then it becomes, let's say, imagine it's a cylinder of the same volume. So when you compress, the size of the cylinder decreases but the diameter increases, and the helicity doesn't really change. But if you do the opposite, if you extend the cylinder, okay, so of course again the diameter decreases, so that's why zDNA diameter is 18 angstroms. But however, the handedness also changes. Handedness actually becomes now left-handed. So how do you explain this? One, to begin with, it's right-handed. then something right-handedness suddenly becomes entirely left-handed. Any ideas, anyone? How this might be possible?
So this probably happens when the DNA opens up or something.
DNA opens up.
Because let's say if you have a right-handed pelvis, you have to make it left-handed, you have to go to this kind of state.
Okay. So, but nowhere actually you have to really open up these two strands. Okay.
So imagine like a rope.
Hmm.
So if it's a coin rope,
Yes.
and you have to make the coin in opposite direction. You have to first uncoin it, then you have to go in
Correct.
opposite direction.
But here you don't have to do. Same thing if you extend
Hmm.
without unwinding.
Yeah.
So it's opposite effect. In B to A you compress,
Yes.
and B to Z you just stretch. You don't have to really unwind. No.
But then how the handle is.
Exactly. So that's the question. Anyone has any idea? How actually a right-handed double-stranded is? Okay. So becomes without really unwinding. One way method we suggest is that completely unwinding. That's possible. Okay. Completely you unwind both strands, and then you wind it in opposite direction. So then of course right becomes left. That's like what you think macromolecular strings, two strings or two threads. But
If the conformation changes around the base of the base pairs, then if the anti becomes syn or syn becomes anti, then
yeah. So who is this?
Sujit, this is Sujit.
Yes, you are absolutely right. So coming back to this structure. So what's important is that you have to keep in mind all these single bonds where actually rotation can occur. Okay, so they are free to rotate. But the moment you rotate at any of these bonds, okay, so very easily, locally, actually, you change the helicity. You change the helicity. Now, if you imagine that happens at every nucleotide, so suddenly you have everything is opposite-handedness, but it's actually occurring because of local changes. And one of the important changes is that if you see the bases like adenine, okay, so one is syn and anti. So basically, you have to really flip. Okay, so sometimes you have to flip. Okay, either you have to flip the entire two strands oppositely wound, okay, from the beginning, or locally you actually change the single bonds. But through single bond, if you flip the bases, so suddenly the handedness is okay. So that's how actually Z form is occurs. Of course, this is not, again, like I said, B form is the most optimized geometry, okay, under aqueous conditions or the cellular conditions. But however, Z form occurs, so whenever you have, let's say, a purine-rich sequence and etc. Okay, so these are anomalies. So you have to keep in mind, okay, so in biology, basically any science. So one is called generalized conditions, but however, you always have exceptions, okay? Given if your generalizations are always basically come from general observations, okay, where there is nothing very highly specific. But let's say if you have, so B form, just for example, if you have here a random sequence of DNA, okay, random sequence, I mean that, so random sequence of A, G, T, C. But whenever you have exceptions, where let's say one base is rich than the other, so then it's not random anymore. Okay, so then of course the specific properties of that base or that component come into picture. So then you have to really keep in mind in finding these structures. Okay, so what are other things which are responsible? Okay, which delineate from one from the other is that yes, the glycosidic bond. So if you see the Z form, so anti for pyrimidines, but syn for purine. So this specifically, this is one of the major changes which allow B form to become Z form. And other minor changes are basically how the sugar. So sugar has 5 atoms. So all 5 atoms are not in plane at any point of time. So at any point of time, only 4 are in the plane. Okay. So one of the atoms, so typically C2 or C3, so goes out of plane, and then again those are responsible for the changes. The double-stranded DNA takes place. So, so this is very interesting. And when we say DNA, typically most of the time we talk about double-stranded DNA, because if you see in the cellular context, so most of the DNA is in the nucleus, it's in the double-stranded form. Only occasionally it is unbound for the gene expression so that the genetic information is translated into something else, and then that will down stick. Okay, so so in that sense we talk about mostly double-stranded DNA, but occasionally so you have to go beyond this double-stranded nature, because this is not a static structure in the end, so you have to transfer this information, right? So obviously you have to disrupt this double-stranded DNA. Okay, either you bring in other enzyme, you cleave locally, or you unwind locally, and then add the information. So then you have to really think about structures other than simply double-stranded DNA. Okay, so that's where actually the DNA triple helix comes. Okay, so
I have one question.
yes.
So in the conformations of A, B, and Z DNA on that slide.
Yes. Okay. Yeah.
So first question is when with the differences of the conformations, thermal stability decreases or increases, means is there any effect on thermal stability?
Yeah. So B is most thermodynamically stable. A and Z are less.
And why is that? Means why is Z less stable? What's the chemical reason behind that?
No, Z is again only it's if you have purine rich sequences. Okay. So then if purine rich sequences take this same conformation, so then Z form is safe, right? Okay, so then again, what is the strength of this stacking is actually important. For example, bases in all these forms of double-stranded DNA, they stack, means that one base is above the other. So you have to really think that how much surface area of one base is actually overlapped with the other, because that constitutes the van der Waals interaction or the dipole-dipole interaction. And of course, this base is stacked with the complementary strand through hydrogen bond. Okay, so this stacking again is actually different in B form versus A form. Okay, so again in B form, if you see the stacks, the bases are actually, the plane of the base is almost perpendicular to the axis, very little tail. So the table shows that 6 degrees. But in the other, okay, so A form, it's more tilt. Okay, so 20 degrees. So basically, then all the planes. So I don't know if you can see my hand. All the planes are not like this, but the tilt means that now the area overlap is actually less. Okay, right, and if you see the Z form, okay, so Z form again it's much different, though the tilt is less, but one base is actually if you see the B form, they actually nice kind of circular staircase. You can really imagine. Okay, so the neighboring bases area is actually much larger, but in the Z form, you see these are like zigzag. Okay, so again actually reduces the area of contact between the bases. So this actually partially affects the stability. Okay, and then because it is less stable, so obviously it's you require less temperature, less temperature. And yes, Suma?
So one more question about that.
Okay.
So sir, is
Yeah,
someone saying?
No, go ahead. Go ahead, Suma.
Okay, so the next question is for the A form, we heard the reason that there is less water content around the DNA, and that's why it's getting compressed. So what's the possible reasons for the Z DNA to be in that form, to allow the NT for pyrimidines and syn for purines, so that it can acquire the left-handedness?
So that's what I said. So it's not that if you have under normal conditions, okay. So if you have a random sequence, so then B form is possible. But imagine if you have really a very high purine rich sequence, okay, and less pyrimidines, and then again this is only short stretches. It's not that the entire DNA will be okay. What's projected here is that the cartoon structure for 36 base pairs, assuming that all base pairs actually form this. You can always have, let's say, if you have a very long double-stranded DNA. It's not that the entire double-stranded DNA needs to be in B form. Okay, locally, let's say a 5 bases or 10 bases can actually get this Z form. And it's possible, like I said, that only local changes need to be occurred to change the helicity on the conditions. So these are actually conditions which are brought in either because, let's say, a double-stranded DNA, zDNA by the way, is actually present inside the cells, but aDNA is not present because most of the time you have actually water, enzymes. Only in crystals you have less water, so that's why A form. So A form, I don't think anyone actually really formed inside the cells, but zDNA is formed. The reasons is that either locally you have purine-rich sequences, are conditions in which, let's say, so these DNA somehow interact more with, like I said, that they have less interaction with each other, less than. Okay, it means that what? So more surface area is available for outside things. So imagine if an enzyme coming in, so then actually it needs to interact strongly with this. So then you need actually more surface area. So zDNA has more surface area for the same number of bases. So this enzyme might bind strong. So then it will actually dictate this structure. So locally you perturb B form to Z form. So these are the conditions which may, so you call in chemistry perturbation. Okay, so local perturbation will give rise to this kind of structure. Okay. Okay. So
I have a question.
Tushar, go ahead.
Yes. So sir, can you please explain why the aDNA has a increased base pair per helical turn while we are compressing it? It is ought to decrease. So why is it increasing the number of base pair per helical?
Imagine this is like a cylinder, okay, and you have a volume.
Yes.
So now you compress the cylinder. The volume doesn't change. So then the diameter will increase.
Yes.
Right. So that's way to explain how the diameter actually increases. That's your question, right?
No. How does the number of helices, number of helical bases per helical turn increases? It's 10.5 in the bDNA, and it is 11 in the aDNA. So how does it increases? It's ought to decrease because we are compressing it.
So compression doesn't mean that you change the helices. So compression means that you have to really rotate, right? So you have to rotate these bases along the axis. Okay. So without, let's say, if you compress without changing the number of helices, so then it might not fit. Automatically, the rotation needs to occur because you don't really change the bond lengths, right? Only the diameters actually will change. Okay, where actually single bonds are rotating. So basically, automatically when you compress, so this helicity changes because of all the characteristics or the configuration. Okay, so where actually the bond lengths are more or less bond lengths and bond angles, okay, are fixed. Only single bond rotations are we call what is called dihedral is what actually changes. So automatically, if you think about that, so that will automatically comes into effect. Okay, because so if you, let's say, compress and you don't want to change the number of bases per complete turn, then you have to really change the bond lengths. That's the only way to it. Okay. Okay. So let's get back to.
Hello.
Okay.
Sir, I have a question that when we can see that we can see that in zDNA the structure is much more relaxed than bDNA. Is it because of the inefficient stacking and the conformational changes which occur in pyrimidine and pyrimidines?
Yeah, so conformational changes are actually basically main changes. Basically, if you see the glycosyl bond conformation, that's actually huge change. The base, which is attached to the sugar ring, flipped 180 degrees.
Yes, sir.
That clearly you can really see in the picture. Okay, so let's say,
Hello?
yes, Shreyas.
Yeah. So does the human DNA exist entirely in the B form?
Yes, so most of the DNA chromosomes, most of the DNA is in B form.
Okay.
Okay.
Right.
Most of it, I would say, I can say 99%, but maybe not 99%. I would say at least a majority. Okay. So when you think about, let's say, yeah,
One is the A form.
Mhm.
You have the base pair difference is 2.6 angstrom. But that really lies in the impulsive region of 5/7. Then what is the extra thing that is giving the A for local setting? It's a local minima, right?
Yeah.
So what is the extra thing that's giving A for its primitive like is the local minima?
So local minima. Okay. So what you see is that. So what you see in the B form. Okay. What is more exposed here? B versus A. You see visibly. You see a lot of gray.
Yes.
In B, right? And less gray in A. So this gray is basically the base pair, means the sideways base pair exposure. That exposure actually decreases. Okay, because that exposure now, imagine partly lost water.
Yeah.
Now, because less water is there, so it wants to increase interactions with itself. So that's how actually it creates. So it wants to seclude itself. So that's the interaction.
And the other thing is the families is pretty. Why she got BDNA and not ADNA? Because whatever the results in DNA, it corresponds to ADNA.
Yeah, that's correct. So then I think we have to go back, and that's a very good question. I don't have an answer. So we have to really see that. So this is a DNA fibers. Okay. So means that these are really large stretches of DNA. Okay, and not really short stretches. Okay, so these large stretches of DNA, and also it might depend on I don't know. So they are not crystals, by the way. Okay, so this is A form DNA is crystalline. Crystalline means the short stretches. Okay, they form really crystalline means that high refined structures, and then higher contacts between the molecules. So in the DNA fibers, so I don't think there are huge contacts between strands, and because these are much longer than that, so that's possibly maybe I think that's a good question. I don't have complete answer, but that might be the reason. Okay. So let's say, so then physiologically it's important that you have to really think about unwinding double-stranded DNA and then bringing other molecules, especially enzymes. Okay, so then so once you unwind, the bases are actually open. Okay, and if you want to, let's say, do what's called DNA polymerization. DNA polymerization. So then you need basically an enzyme which binds a template strand and then synthesizes a new strand. Okay, so then you have a third strand in combination with two strands. Okay, so in that sense, now you have to really think about structures. Okay, so if you have 3 DNA strands, okay, occur together. Okay, so then what might be the interactions? Because then you won't have pure double-stranded interactions. Then the third strand also might interact. That's one of the cases where actually, so interaction of the third DNA strands along with double-stranded DNA might be very important. And second is that just purely playing the structural biology. Okay, so let's say, can you make really new structures? Okay, with existing templates. Okay. So now let's say that's actually possible both in vitro and in vivo. So what is shown in double-stranded DNA? So what's important is the Watson-Crick base pairing. Okay, so that base pairing, hydrogen bonds are actually perfectly stable, linear between donor and acceptor, are Watson-Crick DNA. But in DNA triple helices, means that when a third strand is actually interacting with already existing double-stranded DNA. So what kind of interactions? How these bases actually interact? So interestingly, the slide shows that so where the third base actually interacts, okay, in the plane of already the base pair. So existing base pair. So what is shown on the left in this is the black is the thymidine, okay, interacting with adenine, 80 base pair. So this is Watson and Crick. Everyone sees my arrow?
Yes, sir.
Okay, okay. So this is Watson-Crick base pair. Okay. So where A and T, we know that how many hydrogen bonds? 2 hydrogen bonds, and these are actually perfectly linear. Okay, very stable form. And if you bring in a third strand, so in this case, the base coming from the third strand is again thymine. So this thymine actually now can interact with adenine already in the double-stranded DNA without really affecting this hydrogen bond. It hydrogen bond with Watson-Crick base. And again through hydrogen bonds, because this adenine has another hydrogen atom on nitrogen, which can actually act as a donor. And this hydrogen on this macrocycle again can accept hydrogen. Okay, acceptor. So because of this donor and acceptor, now can pair with another P which has one donor and one acceptor through hydrogen bonds. Okay, very similarly than Watson and Crick. But what are the differences? Can anyone point out differences between AT of Watson Crick versus AT of triple strand? Crick versus so this is called Hooke's scheme. Yeah, Hooke's scheme. Okay, Watson Crick pair versus Hooke's scheme pair. Okay. So normal way of hydrogen bond is called Watson Crick, and these hydrogen bonds, okay, are called Hooke's pair because these atoms are often called Hooke's atoms. So this nitrogen on adenine, okay, is a Hooke's and also I think sometimes even this nitrogen oxygen also called Hooke's. Okay. So can anyone point out what are the differences between Watson-Crick pair and Hooke's pair? Hooke's pair is completely on the pink between A and T, and Watson-Crick is on pink A and black T. Anyone wants to highlight what are the differences? If you have to write, looking at this structure, differences between Hooke's pair and Watson.
Here the nitrogen from the 5-membered ring is being involved in hydrogen bond.
Okay. Nitrogen from the
5-membered ring of the base.
Okay, but that nitrogen is involved in which base you are talking about, Shubhra? Which base you are talking about first, because there are a lot of nitrogens.
It says
So
that,
Adenine.
that adenine with it.
Adenine.
Okay, adenine, nitrogen. Okay, so that's new. Okay. So what is in terms of hydrogen bonds? Are these hydrogen bonds are similar to the Watson-Crick hydrogen bonds?
Sir, maybe in the Hooke's pair, the angle the angle between on the one base with the other, the hydrogen bond can rotate.
Rotate, hydrogen bond rotate. Okay.
Means the
So you can say they are not linear. Okay. In
yes.
Watson-Crick, perfectly the donor atom, acceptor atom, and the hydrogen all are linear. Okay. So when you say these hydrogen bonds are linear, they are really strongest. Okay. So here, neither bonds are actually linear because they can't be linear, because if you see again the bond distance, bond lengths are fixed and the orientation. So whatever orientation you bring in, this is the best orientation to bring closest to the linear or reduce the angle so that it becomes as close to the linear as possible. Okay. So this is because the third strand here, what is shown is that only 3 bases in a plane. But now you can actually really extend if you have more bases, or the length of the DNA strands are long. Okay. And similarly, example shown on the right side is for, so this is for what is shown, so T, okay, often shown. So typically the nomenclature is that if you write this double bonded sign, that's a Hooke's that's Watson-Crick pair, and typically put a dot just to differentiate that. Let's see Hooke's pair. Okay, so the hydrogen bonds is actually hydrogen bonds, the structures are different. Okay, H bond. Okay, structures are different. Okay, and of course, the atoms which are involved are also different in some ways. Okay, so on the right side, so another Hooke's A. Okay, so GC is a normal Watson-Crick pair, shown again guanine in pink and cytosine in black. So that's the normal Watson-Crick pair. Okay, in BDNA, all the hydrogen bonds, okay, all three hydrogen bonds are perfectly linear, very high. But if you see the other cytosine, okay, which is shown in pink, again has two hydrogen bond donors, okay, which can donate hydrogens to two acceptors and one. Okay, again, can someone point out what are the differences between Hooke's pair versus Watson-Crick pair in C, G, C, K?
So here we have taken cytosine.
Yes, Ashwarika?
We have taken protonated cytosine in Hooke's base pairing.
Okay, very good. So here, the cytosine is actually protonated. So there is a hydrogen atom which is added to a nitrogen, so that which can act as a donor. Okay, so without that, so you cannot really have Hooke's pair between C and G, because you need that positive charge. Otherwise, you will be able to give only one hydrogen bond, which is not really stable. Means that, so if you have the protonated C, so then it can form very easily Hooke's pair bond with this. Okay, that's one aspect. Okay, what are the other aspects? Again, a similar aspect. So the linearity, the strength of hydrogen bonds. So again, both the hydrogen bonds are not linear, as in Watson-Crick GC. So and also again only two hydrogen bonds. In Watson-Crick GC, you have three hydrogen bonds. So these are not really as stable as this. Means that the third strand, okay, the strength of third strand binding to double-stranded DNA, the third strand is actually not as stable as this. But yet you have three strands come together to form what is called DNA triplets. Okay. What is shown at the bottom is a structure of, if I remember correctly,
Would it be, is it okay to comment
yeah,
that in presence of a Hooke's pair, the energy stabilization of the Watson-Crick base pair decreases? Because I can see like common donors and common acceptors.
maybe very little, I would say. Would not really, as long as, okay, so then you have to define hydrogen bond carefully here. Basically, how much of that hydrogen is actually shared to acceptor. So if that strength is, I mean, if the, of course, if that changes, the strength of the hydrogen bond changes. But I don't think it would change dramatically. Okay, so very small extent, if at all. Okay, so no, it should not change too much. Okay. So,
Sir, I have a question. That
yeah, Sujit.
Sir, why system is allowing the third strand to bind with the Watson-Crick pair? We have a double strand DNA, so we have the third strand is binding with that. So why system is allowing to bind the new strand with the already existing double strand?
No, to stabilize its interactions. So why something binds A binds to B, because the binding stabilizes. You know, let's say the binding releases energy. So
sir, already we have 80 base pairing, stable base pairing. So why
see, additional hydrogen bonds are coming, right?
Okay, sir. Okay, sir.
So without disturbing the original, without changing the energy of the original double strand, you are bringing more stabilization. Okay, so the same question you can really ask: when two strands get separated, why they bind to each other? Because through the hydrogen bond, other interactions, they are stabilized. So third strand actually is bringing stabilization. So only again you have to see that when these possibilities occur, then only. Let's say, for example, 80 is there. So if you have another T, then only it stabilizes. Okay. So like this, you can really think of. I think the cartoon structure below here shows a triple DNA triple helix containing, I'm sure, many bases. only the colored one is actually one triplex. Okay, so in that axis, you're seeing the structure from end on or from the top or bottom. And what color is that? Only 3 base pairs. Okay, so which are in a plane forming a DNA triplex. Okay, our base triplex is shown here. Okay, so the now question is that can we add actually more strands to stabilize the DNA? Okay, so 3 bases, it's very interesting. 2 is already interesting. Beautiful hydrogen bonded structures, very stable. And when third base comes in the plane of 2 bases, again we have seen very interesting structures. Okay, TAT or GCC plus.
Sir,
Yes.
so this structure has also been deduced from the X-ray crystallography, or?
Yes.
Okay. So then another question is why the another nitrogen is not bonding with hydrogen to form a positive nitrogen so that the hydrogen bonding can occur.
Which nitrogen you are talking about?
The 5-member nitrogen. Why it is on the 6-member nitrogen the positive nitrogen plus hydrogen is forming?
So you're asking that in CGC,
Yeah.
why the positive charge is on the
Yeah, because the hydrogen bonding can also be happen with the hydrogen is shifted to the another nitrogen, and then the same thing will happen with this. So is there any?
So then actually you are changing, right? So if you change now one hydrogen, if you put it on, let's say, normal GC pair, so then basically it's a protonated, let's say, protonated guanosine. So then that actually changes the entire. So you're adding whole positive charge on the original guanine, which is actually stabilized by hydrogen bond with original Watson-Crick strand. So that also might have huge effect now because you have a huge positive charge. But however, you won't affect that much because the third strand is affected because of the protonation. Okay.
Because this structure had been reduced in the X-ray, so you can say that H positive is exactly there.
Oh yeah. So you can use one. Okay. And you can use NMR. So NMR will give you beautiful.
Yeah, exactly.
Change in the environment gives rise to very small sensitive information about this. Okay. So,
sir, I have a question that is when in our system double helix structure is natural in our human body.
Correct.
So when in which conditions system is allowing the third strand to bind with our double strand, or it is artificially made?
So, well, these are artificial structures, but they might occur naturally. That's what I'm saying. So, as long as if you have third strand which is available, okay. So it doesn't have to see. You don't have to really think all the time. When I say triplet, it's not that huge three very long 3 strands come together and bind. Even if you have few base pair strands, or sometimes it's called oligomer.
Okay, sir.
In an oligomer means that a few few nucleotides. Okay, imagine let's say like endpoints. So you can call it nucleotide or oligomer. So these oligomers or oligos can exist. Okay, if they exist, and then if they are double-stranded DNA available in the neighborhood, then they want to bind so that they can stabilize and reduce the energy. Okay, in those circumstances. Otherwise, naturally you have in chromosomes double-stranded DNA. Okay, double-stranded DNA packed with proteins and what. Okay. So,
sir, I have a question.
Sir, I have a question.
Yeah, make it short. The class will go for
So I am going first. Okay, sir.
Yeah.
So I am telling that so when the another base, that is thymine, bonding with the AT base pair.
Correct.
So for this the other adenine, which is upper side with the thymine, so those hydrogen bond will be breakable because if this thymine bond with the hydrogen bond with the AT base, so there may be some distortions on the DNA will be occurring because if 3-3 type going on, so the distortion in the DNA can I mean to say that it will be stable or it will more collapsed or otherwise it will spread many more like elongated or constructed. I want to say that because.
Okay. See, what you have to really simultaneously think that here we are talking about only the interaction between the bases, and all bases are in one place. Okay. So, but this nucleotide has a backbone, right? You have sugar, you have phosphate, okay, etc. So again, you have to really see that how that also affect the structure. So mind you, so triple helixes are not really very long. Okay, so the third helix is typically very short. Okay, interacting with locally. So then locally it might have very small differences. Okay, imposing on the double strand, but not, it doesn't really change dramatically because overall it needs to be stable. Okay, if it, let's say imagine the energy minimization the third strand brings in, okay, should be much higher than, let's say, if you have to really put a little bit strain in the original double stranded DNA. So it will not really affect the double stranded DNA, but it will add overall stabilization. Okay, so in short, so it doesn't really have huge effect on original double stranded DNA structure. Okay, but locally it can happen little changes. Okay, for example, like ADNA, what we have said that basically when less water is there, so you want to really have bases stack much more efficiently. So reduce the surface area. So like that, if the third strand is coming, okay, so to have better overlap between, let's say, all three bases, small changes might be. Okay, so moving forward. So we know that now three bases can really form actually planar structures within DNA strands and form what are called triple helix. Okay, which might have applications, which might have implications both in vivo as well as in vitro to make interesting structures for different reasons. Okay, so is it possible that four bases can come together and form interesting structures? So interestingly, so in chromosomes, okay, so chromosomes is where we have our DNA material is saved inside the nucleus, along with, of course, a lot of proteins. And if you and these chromosomes, if you move towards the end of the chromosomes, okay, so there actually sequence is not random anymore. Okay, it's not random. Random, I mean random in the sense that, of course, genes have a specific sequence of DNA, but still within that, it's basically the composition of A, T, G, C, more or less random, more or less equal. You don't ever really have. But towards the sequence is not random at ends. Okay, at ends of the chromosomes. Okay, so if you see these sequences, now we have genome available. Okay? But otherwise, even before that, people know that ends of the chromosomes have actually very peculiar DNA sequences. Okay, so one of the peculiarities we have found is that this is guanosine-rich sequence. Okay. Okay, so these are G-rich sequences, and typically people found that these sequences themselves are actually forming structures. Okay, so without having a complementary strand. Okay, so if a see what kind of structures it forms. So now, so four bases. Okay, so four bases can come together, and then they form hydrogen bonds. Okay, so with each other. So the structures I'm showing here is that, so one of that such plane where actually four bases literally form a plane, and then the interactions between the bases are actually stabilized by two hydrogen bonds. So typically guanosine gives 3 hydrogen bonds to GC. Okay, GC, right? And these hydrogen bonds are perfect, you know. But if you have 4 guanosines, interestingly it forms what is called G tetraplex, we call it quadruplex sometimes. And you see what you see on the left side, the beautiful structure of all these 4 guanosines, okay, hydrogen bond with each other. And of course, these hydrogen bonds are not linear as we know, but still, because 4 bases are coming together, form interesting quadruplexes. But how these 4 guanosines come? Are they 4 different strands? It's possible. In vitro, you can actually have 4 different strands and then can come together and form this. Otherwise, what I am showing at the bottom right, you see a single sequence, okay, single standard DNA. Imagine a lot of G's are there, okay, along with other bases, okay, which are in the loops, can form this kind of tetrad. You see the 5' to 3', it's a single DNA sequence, single strand sequence, can form such complex structure where you can really have, for example, in this cotton structure, there are 3 stacks where actually you have 4 guanosines come and bind together to form this kind of structures. So these are called guanidage. And initially, people found these sequences only in vitro, but then later we realized that these sequences are actually really present in chromosomes themselves, and these structures are actually very, very important. This is one of the ways actually the DNA itself is actually having a stable conformation. Okay. So that is a G, okay, and what is shown in picture D is basically how different stacks. These are real structures, okay, derived from NMR, okay, showing that how these G's can come together, okay. So in double-stranded DNA, you know that both strands are actually anti-parallel, right? So in one case, it's one strand goes from 5' to 3', another strand comes from 5' to 3'. Okay, so anti-parallel BDNA. Okay, BDNA, ADNA, ZDNA, all are anti-parallel. But in this case, you see G tetraplexes, only bases are actually forming strands. But how these bases are actually arranged? Are they these individual strands are parallel or anti-parallel? Different possibilities actually really bring this. Okay, panel E shows that you can have parallel strands, all 4 parallel strands come together. Okay, which could be again really coming from a real single stranded polynucleotide, but it could be different strands. But the stack will have all 4 parallel, or it could be anti-parallel. Okay, this is interesting. So you need to realize that these structures are really possible. Okay, so once I show G tetraplexes, what are the immediate questions arise to your mind? What is the next immediate question will arise? When I say that G forms, okay, a stack of 4 G's, you can form tetraplex.
C and T also may form such thing.
Someone needs to speak loud. Yes, Sanjeev.
And C and T also may form something like this.
Yes. Okay. Immediate question is that obviously you have 4 bases A, G, T, C. What about other bases? Okay. So G is of course popular because it's really found because you have a lot of G sequences in chromosomes. Okay. So physiologically very important.
So I have a quick question.
Yes. Shuman, yeah.
So is this natural? This bonding is this found in? Is this natural? Means nature had such organizations, or it's in lab in vitro?
No, that's why actually the sentence I have here telomeric region at the ends of chromosomes. So this is in your body you have G sequences. If you see the sequence at the ends of chromosomes, there wherever you have the G sequences. Okay, you don't need a complementary strand. Okay, more or less, the single strand itself can come and form this. And yes, this is natural. And for your information, there was a Nobel Prize which was awarded for actually recognizing, detecting telomeric regions in inside human DNA, and also related enzymes and how it might be even important for cancer. Okay, so there was a Nobel Prize award about maybe 10 years. Okay, in last 10 years. So yes, immediate question would be, okay, why not other bases? Can other bases really form? Okay, I'm really proud to show you this because we were the first ones. So this was G tetraplex. I think early 1990s. People actually looked at this, and then as a natural question, everyone really thinks otherwise. So then actually, as I was a very young graduate student like you, in my first year itself, when I was actually doing a project with Professor Huzur, okay, such a biologist and I'm an expert at PIAPA who is superintendent now. So when I did a project with him, so we were asking a very simple question. Okay, G forms, why not A? Can you really make A? So we have actually really shown experimentally NMR through NMR shows that if you have you can have 4 adenosines coming together and form tetraplex just like G tetraplex. Okay, what's the stabilization? Hydrogen bond. Okay, so all 4 bases in the plane. Perfectly similarly, you can really think of what I'm showing in A and B are actually 2 slight variants of tetraplexes, but formed by AAAA. Interestingly, beautifully formed. Again, each one gives 1 hydrogen bond, unlike in G where you have 2 hydrogen bonds. That's one of the differences. And depending on which donor, you have to look carefully. The donors are slightly different in A and B, but nevertheless, the structures are slightly different, but these are related. Okay, so this is the manual roughly about 20 years. Okay, this is my first year project B. Okay, after lab rotation. Okay. So this is my first research piece of work. I was so excited, and one of the well recognized work. Okay, so then you can really think of other tetraplexes, right? Of course, Professor Hosur actually found out 3 tetraplexes. Okay, again, all T's, if you can really put together, they form. Okay, so shows that, and of course, just to complete the story, so C also can form tetraplex. Okay, C again, single hydrogen bond. Basically, base to base, it's the single hydrogen bonds. Of course, some metal ions are required sometimes to stabilize. So what it shows is that, so all these things, so depending on what sequence you have, whether you have a complementary strand or not, you can really have very rich structures just based on DNA. Okay, we began to think, okay, DNA means just double-stranded DNA. Okay, yes, most of the chromosome you have complementary strands perfectly paired DNA. But however, so whenever you go to sequences, okay, towards the end of the chromosomes, where actually you don't have to really have a complementary strand all the time, but the strand itself can form really rich structures. Okay, so imagine if something is G, complementary strand we have C. Okay, so either you can have GC double-stranded DNA, but otherwise G itself actually able to form tetraplexes. C itself is actually formed to tetraplexes. Okay, so these are the state-of-the-art questions one needs to really ask, and of course they have been asking. But showing this in vitro or in vivo, okay, so are detecting, okay, so will really help you push basically understanding the biological systems. Okay, so all this work, because it's possible, not only studies directly looking at natural systems, but trying your experiments in test tube and then say that, so in test tube I can make this. Obviously, nature has tried over millions of years. Why not? Might be possible there, but you need to go and detect it. Okay, so that's the crux of the story. Okay, but we have learned aDNA, bDNA, zDNA. Okay, so there is something called sDNA. Okay, so this is called a stretched DNA. Means that if you take a double-stranded DNA, yes,
so in telomere region is a G-rich region, and you find a lot of G tetraplexes.
G tetraplexes.
You should ideally find an equal number of C tetraplexes.
Exactly. So, but I don't know how in vitro one might actually detect. It has to be, because finally the chromosomes, right? So obviously you replicate as a strand. So obviously you replicate even G, means that you replicate with C, right? Obviously DNA polymerase doesn't really stop. So you're absolutely right. So if there is G-rich, that's why I'm talking about. There is G-rich, complementary strand should be C. So G itself is forming tetrad, well known somehow, but C also should be able to form. Okay, given the conditions. Okay, the important question is that are G form relatively? What is the population of GC forming a Watson-Crick, the bDNA? Quadruplets. Yeah. Under what conditions the transformation occurs? Okay, which one is more important for the cell? Okay, so there might be reasons for which they want to have single-stranded segregated. but clumped together then form these structures. Or maybe single stranded together, they bonded together. So these are the important questions we don't really know the answers. So these are the things we need to really go and pursue this. And one of the ways actually, how do we know in nature, inside your cells, gene edits are possible? It's not that you can study right away chromosome, right? So what people have done is that, so we also actually, I did some work is done. So how to detect G tetrad versus let's say a natural DNA. Okay, so there are actually you develop.
Excuse me, sir.
Yeah, just a minute. Let me finish this. So you develop probes which can detect very selectively G. Okay, imagine if this structure is possible. So you can have fluorescent dyes. Okay, which actually bind only to G tetrad, nothing else. Okay, on the sideways, for example. So once you develop these tools in vitro, you add to directly to the cells. And then if it goes and binds, and if you have the probe properties like fluorescence intensity, lifetime, you name it, they're identical, then you know that very likely inside the new chromosomes we have. So that's how to detect. And after that, I haven't really followed much because I switched my fields. People have actually developed fluorescent probes which detect C tetrad or A tetrad. So now you can actually add these guys to your cellular pool and then ask questions that, okay, so how much of my DNA is in this form? Okay, so these are the state-of-the-art questions. These are very relevant.
Sir, I have one question, sir.
Sanjay, just a minute.
Into the metal, it should be much easier to detect.
No, okay, so no, I didn't really go to the complexities, but these structures, you need actually, you see the central hole?
Yeah.
Okay, so central hole you need to fill with something. So most of the time it's potassium.
Potassium.
Okay, K plus. K plus actually fixed in that partially stabilizes. Okay, so like that, all tetrads you need ions. Okay, so I didn't really go because it's otherwise it will become research talk. Okay, okay, but that's the important question. Yeah. Someone had a question, student.
Sir, I have.
Yeah.
Sir, while reducing the structure of alpha helix, sir, because the composition of G is equal to C and A is equal to T, we decided that there is a complementary base pair because only because the composition are same, only because of that data.
Yeah.
So here we are seeing that G and G is also forming. So then definitely, if we take a concentration from a DNA, means nucleus, so definitely G will be more. So where that the data come from that G is equal to C and A is equal to T?
No. So we have to keep in mind. So what I'm showing that triplets are the quadruplets. This is not the canonical structures. So most of your DNA in chromosomes is double-stranded and complementary strands. So what we are talking about, so let's say imagine that's why I mentioned G is very importantly towards the end of the chromosomes. Okay, so we have how many? 23 pairs of chromosomes, right? So if you go to the ends of chromosomes are really long, right? Millions of base pairs. So towards the end of the chromosomes, you have purine-rich sequences like G-rich sequences. So what we are talking about, so let's say if you cut those sequences and put in your test tube, so what structures do they form? People have found both in vitro and also detected in vivo. So there's a possibility that they can form this kind of rich sequence. Okay, but this is again richness is important. You know, again, don't think that any random. It's not just 4 G's come together. So what's important?
So, are these forming the these G stacking are also in the helix kind of thing?
No, helix. What do you mean helix? So, well, 4 strands come together. So there is a angularity handedness. Yeah. So what I'm showing in the D, I think it's if I remember correctly, how many stacks? Most probably 3 stacks. Okay. So there is angularity. You know. So the small and helicity will be there. Either handedness. Small handedness will be there. But don't think that this will go forever. I mean, it's not like the stacks which are shown in the cartoon, let's say 3 tetrads, right? It's not that it will form 10 tetrads. Okay, you won't have that, the long sequences. Second thing, so you have to 4 strands come together. Okay, so the
Okay.
cartoon structure at the right bottom shows that, for example, the loops. Okay, we didn't really write anything in the loops. So it's not that the loops have to have some bases, right?
Yeah.
So those bases actually will not be able to satisfy your hydrogen bond. So like that. So it's not that this is not the canonical thing, but these are only towards the ends of the chromosomes. Okay. Yeah. Someone else wants to ask a question?
Yes. So my question is that so why is the telomeric region G rich? It could be A rich or T rich. So why is it G rich only?
No, that's what I
Or
was.
else-
That's what I was addressing a little bit when he asked. Okay, so most probably you have a complementary style which is C rich. Okay, but traditionally for some reason we found out that G rich sequences. Okay, so if you have to, let's say, have exact complementarity in copying chromosomes, okay, with high fidelity from the first base to the last base, then you should have complementary C strand, okay, and in vitro studies clearly show that C also can really form tetraplex. So obviously the question is important that C also might be formed, okay. Okay, but those are nitty-gritty details. Basic bio. Okay.
Sir, also a small question. Sir, sir, now can possible that the pentaplex, hexaplex, all possible? Why only?
No. So you have to keep in mind that now, let's say if you have, let's say 5 pentaplex, okay. So what happens is that now this is like crown ether, right? Kind of the ring size. So if I have to really think about a ring, okay, 4 bases form, let's say square, okay, 5 bases form, let's say pentagon. But then you need to stabilize these structures. Okay, so what I didn't really mention a little bit more details of this. So these G tetras, okay. So okay, what is shown here in the C at the center? You see what's the ion? You see at the center?
Yes, barium 2 plus.
Exactly. Okay. Means that so you need something else also stabilizing because each base is doing only one hydrogen bond donor and one hydrogen bond acceptor. Okay, this is not the super stable structure. Means that you need some more stabilizing element. For example, in this case, in vitro they put really barium and stabilize, but naturally you have potassium. So potassium or sometimes sodium, I think, can actually stabilize. So all these tetraplexes so have a central hole, if I can call. Okay, for example, G is very clear. So central hole, you need to put a metal ion, okay, to stabilize this stack. Now, if you have to really imagine, let's say a pentagon or hexagon, then the central hole is really large enough that you can't really put one metal ion stabilizer. So I don't think they will be very stable for the reasons of many strands come together and stabilization force which is provided by metal ions will not be present in larger things. And to my knowledge, I haven't really seen anyone finding pentagons or pentaplexes or sexaplexes or septaplexes and so on and so forth. Okay. So to finish this, okay, what are the different forms of DNA? So there is called stretched DNA. So this doesn't really occur by naturally itself, so you have to really follow. So for example, the kind of experiment which we do in our lab, where we use force spectroscopy, where you can really apply stretching forces to different biomolecules. We do in the lab almost all the time to protein molecules. But similarly, you can really apply the stretching forces to DNA, okay, by methods such as, let's say, optical fuses. So how do you do this? So you take literally double-stranded DNA and attach both strands on one side and other side to something, and then imagine you begin to stretch. Okay, so once you begin to stretch, so then of course your extent. Okay, so whatever the energy minimization provided by base stacking and the hydrogen bonding now going to be disrupted. Okay, of course, if you make it really longer, both strands will have now they will lose the helicity and they become linear, right? So then you will not have perfect hydrogen bonding. So this is called kind of mostly melted as single-stranded DNA. So what is shown on this picture on the panel B? Okay, panel A is basically a cartoon picture. Don't worry about the details. So imagine that you had a DNA on both ends and then stretched. That's all. On the panel B shows that as you extend from the natural length, as you extend, okay, and how much actually force is required to extend. Okay, so it shows that initially actually it goes very nicely. Okay, so basically proton structure, maybe hydrogen bond is slightly off, base stacking goes off because you're rotating and the helicity changes. But then after certain strength, if you stretch further, base pair they break. They break and they give you nice elongation. Okay, so this happens base pair by base pair, and then until all the bases one by one break hydrogen bonds and become single strand. So once they have become single stranded, so then you are stretching both single stranded, which have different what are called force versus extension behavior. The picture nicely shows that initially the double stranded DNA, where the force increases, but the extension doesn't really change. You know, they extend very nicely. But there is a plateau occurs at 65 piconewton shown here. At 65 piconewton, you will keep stretching, the extension goes a lot, but the force doesn't change because you are extending each base pair will give you a little extension, but at the same force. Okay, this is called cooperative transition. But once all bases are extended, there is nothing else to play, and then single-stranded DNA again will show slightly different. Okay, so this is called sometimes B to S transition. Okay? So this is a kind of well-known transition now, BS transition. Okay, in DNA space. So double-stranded DNA is not single-stranded DNA. Okay, double-stranded DNA. So you reinforce the spectroscopy. I can really talk on this for hours. Typically, I teach this in single molecule biology course for spectroscopy. Okay, so you can really, appreciate that even the complex tools which we have developed over past a decade or two can be really used to actually address fundamental questions like this. Of course, this melting, this is called force-induced melting or mechanical stretching melting. Of course, it's artificial, but to mimic why these experiments are important is that. So this is how exactly the melting occurs locally when an enzyme comes. When an enzyme comes, literally it needs to stretch. Okay, so one of the ways is that. So you can now imagine that locally if it is extended. Imagine, let's say, if it is this kind of extension occurs in this experiment over, let's say, 100 base pairs. But an enzyme can do this locally at one base. Means that one base hydrogen bond you need to literally break. Okay, and then now that basically the unwinding occurs locally, that can be propagated along the strands so that the strands actually melt locally, now available for other enzymes, for example, DNA polymerase can make another copy of DNA or RNA polymerase come on bind single strand DNA to make RNA. Okay, so just keep in mind that this is kind of, if you are interested, you read these tables. Okay, one is, I think, molecular simulation showing real structures, what happens at atomic level, atomistic level, how the melting happens. The other one is real experiment shown. Okay, so to complete.
Sir, in the previous slide, there was a dip in the curve. This dotted part, can you see that? This dotted, yes. I cannot show you right now. In this graph, so there is a dotted part, not the green one, the black one.
No, I see. So I don't know. I need to really see that. Read the paper. I don't remember right away. But there is, so this figure is taken from this paper. So this is figure 2, I guess. Okay, in this paper. That's why I've given you the reference here. So this is nucleic acid research 2012. We have all the information. Okay, you can actually go on this. But I'm sorry, I cannot recall right away what is that small thing.
Okay, sir. Okay.
Most probably. Actually, I can really tell. So this is it reversible? Okay, so this is an out of equilibrium experiment we call. Okay, so basically you are taking a naturally existing sequence and then you're extending, right?
Yes.
Okay, so it's actually this is forward reaction, imaginary extension. Okay, so you take all the way to let's say 100 piconewton, then you have single strands. But what happens actually if you reverse the reaction? Obviously now, if it is completely reversible, you trace it back. All the processes which are occurring along the forward reaction, exactly replicating the in a reverse order in a reverse reaction. What might be happening there is that, so the moment actually you relax, okay, so there is a snap, okay, so basically this is kind of cooperatively, most bases which are actually still outside suddenly come back and then form hydrogen bonds. That actually even reduces extension and also reduces force. Most probably that's where I would explain.
It's basically a plastic deformation kind of thing.
Yeah, you can really call. You can really call. And obviously, these experiments always, more or less, they show the forward extension and then the relaxation. And always they want to really look hysteresis. Okay, any change in the process in the forward if you reverse the reaction. Any differences in terms of area? So force versus extension. So force dimension multiplied by distance is energy. So any deformation in hysteresis will give you basically energy dissipated. So basically you can really think in terms of hysteresis. Okay, I think that's the answer. Okay, but I'll look it up. Okay.
There is no arrow, no? Actually. So
yeah,
there is no arrow given. No? Is that that's my doubt, because in general in hysteresis graphs we have some arrow of.
Oh yeah, that's true. Yeah, in aggregation, etc. That's true. But here, yes. So there are two distinguish that they have put thick line and dotted line. I'm sure the legend is not present. So in the legend, they might have really explained what is the thick line and the dotted line. But most probably, I think this is the case. Okay.
So one is that if we do the main experiment, same experiment really smooth.
Yeah.
Do we expect the transition to ZDNA?
ZDNA.
So that's much longer. That's the local minimum. If I think only in terms of the extension.
Possibly, yes. Yeah. Yeah, it should be possible. Okay, so yeah, but this is really large. S is really large.
Yeah, it's really large.
Okay. That's a good point. Yeah. Okay, so so far we discussed a lot about the DNA because the reason most of the things you can really predict for DNA structure is that because you always have complementary strand and it's mostly pDNA. So once from 1953 the paper model presented, we know right away for any double-stranded DNA, people say that if they give you a sequence of one DNA, you can predict the complementary DNA sequence, and you can actually predict the structure because you know exactly G is paired to C and A is paired to D. Perfect, right? So DNA structural biology is mostly known except for I mean, let's say triple helix or quadruple helix, even which we know a lot. But what happens to RNA? Okay, so first of all, what is RNA and why RNA is important? So DNA is mostly restricted to nucleus. Okay, the problem is that in eukaryotic cells, so even prokaryotes, so DNA is mostly restricted to the center of the cells, to nucleus in eukaryotes. But it carries the information. Okay, so it cannot by itself go everywhere and then transfer the information. Okay, so you need a messenger. Okay, so you need a messenger which carries the information from the DNA to everywhere else. So typically to generate proteins. So that's how actually another molecule came which carries information from DNA Okay, so whatever information.
Can you repeat why that messenger RNA is needed, sir? Please.
So that's what I'm telling you. So DNA is mostly restricted to nucleus. Okay, and it's basically if you have to carry, okay, so let's say, and DNA is very long, you know, it's not that each gene needs to be expressed every time. Okay, so only certain genes are expressed. So now either you have to do that, the entire chromosome should float around the cell, it should be in extended form so that all genes are accessible. Then the cell size cannot be really hundreds of microns, you know, you need to really have, I would say, maybe millimeter long the cells. So but to overcome that, cells have actually developed a mechanism that how to transmit only the information which is required, okay, from here. Imagine this is like a library, okay? So imagine library is full of books, all subjects, everything. But it's not that every book, every page, every sentence, every chapter needs to be available everywhere in the institute or everywhere in the world. No. So you go to the library and then say that I need information from this. So you go to that book, that chapter, you get the information. Okay? And then you come back with information. So now the cell, so basically DNA kind of library at the center. So it needs to express only certain genes. So then you need to have molecules which convey this information. So DNA cleverly designed this RNA molecule. So DNA RNA polymerase. Okay, so RNA polymerase carries that information from DNA, whichever part, in terms of mRNA. Okay, so initially we discussed what is the difference between DNA and RNA. Is that only two differences. One at the sugar 2 prime, there is a deoxying DNA, but there is a OH group. Second, in terms of is ATGC. Okay, so A is A, G is G, C is C, but in the place of T you have uracil. That's all. Okay. And second thing is that DNA is double stranded. Okay, so that's why most of the time we know the structure. We don't have to really worry about once what's encrypted there. But messenger RNA need not have complementary strand. Okay, more often doesn't have complementary strand. Actually, you can think the same thing. DNA doesn't have complementary strand, but is a structure. You know, we never really bother about that because our natural DNA is double stranded, so DNA structure. But DNA, if you don't have a complementary strand, so then of course this is kind of G tetraplexes, A tetraplexes, these might arise depending on their weight. But if they are not random sequences, then single stranded DNA you can't really predict. Okay, so now the question important for messenger RNA, it's copied directly from double-stranded DNA, but only one strand is copied. Now it doesn't have a complementary strand, okay, most of the time, because it's not required, okay. So only one single strand is important. So this single stranded now can have a rich variety of structures, okay. So same strand can copy each other. So imagine if you have very long strand. Okay, A, B, C, let's say C, 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 C. Okay, so then let's say again you have G, C, A. Okay, A. So this is kind of always remember writing 5 prime and 3 prime means that this is a sequence. Okay, so so in RNA we don't have T, so basically U. All right, and we know that G, A loves to pair with U, G loves to pair with C. Because you don't have a complementary strand, so the strand itself can really form hydrogen bond. Okay, so within the single strand. as long as these bases can come together. Okay, so this, once it pairs, it forms actually kind of, let me put it. Okay, so C, G, U, A. So here it is U, A, C, G. So now, so this kind of structure can form. Okay, so 5 prime, 2 prime. So this is called hairpin. So this is true for single-stranded DNA as well. So these kind of, now if you have sequences, okay, which have complementary stretches within the same span, they can form rich hairpin structures. So what I'm showing you on the slide is the cartoon structure, not cartoon structure. What is a possible pairing occurs in an RNA called M1 RNA from enzyme component of the enzyme RNA of E. coli. Okay, so this is a real RNA sequence. You see very long, single stranded on the right side, and a lot of hairpins are there. Some are long, for example, shown 15 base pairs, 6 base pairs, 8 base pairs, etc. And it could be as short as this. So this RNA structure is impossible to predict even today. Okay. Both RNA structures and protein structures are not easy to predict. Okay, how do they form? Okay, which part of the RNA stabilizes what? RNA is relatively easier than proteins. Proteins are much more complex because you have 20 varieties of amino acids. But here it's nevertheless just 4, but nevertheless is very complex. Okay, so what kind of hydrogen bonds? So sometimes you can have hydrogen bonds like what is shown at the bottom left, GA. So typically G wants to pair with C, but in this structure it shows that sometimes it can even pair natural. So this is natural. So this is kind of forced. We can say. Forced means that. So imagine, let's say, if I have a sequence here, U. Okay. So because this is a good pair, this is a good pair, this is a good pair. So now, so this G and U is forced. Okay, which form hydrogen bond shown here, not a perfect hydrogen bond, but because on either side you have a very stable sequences, so they can also form. So these kind of structures are possible, okay, which might not be, you can really call this is non-Higgs, okay, non-Higgs. Okay, so this is Watson-Crick pair. So this is a non-Watson. Okay, so because of this nature, so even today, RNA structure is a big field by itself because it's not very easy to predict the RNA structure. Okay, so that's mRNA. Okay, so but you have similarly the RNA. Okay, okay, so there is something called tRNA. Transfer RNA, so which brings bases, amino acids. Brings amino acids for protein synthesis. So also have very complex, because again these are single stranded. Of course, conjugated to amino acid. Okay, so they have very similar structures. Okay, so it's impossible to predict these structures, but this base pairing within that sequence are important because it shows that which parts are accessible, not hydrogen bonded, how that might be help in binding, let's say, other molecules like proteins is intense and active. Okay, I think that concludes basically polynucleotides, structures, and etc. Okay, so most of the stuff I have taken from chapter 8 of Lineage. So a lot more is that. Lineage is really a big book, but you can actually read this chapter. Next class onwards, I'll introduce maybe amino acids, maybe just connection from DNA to proteins, and then we will begin protein molecules, amino acids, properties, and intermolecular forces.
Sir, do we need to go through the central dogma part?
That maybe I'll introduce next. That's why I'm telling you, the connection between DNA.
Yeah.
Maybe I'll give you briefly central dogma. I mean, already I have given half the central dogma, DNA to RNA. So then I'll explain RNA to protein as well. Maybe I'll give you, I'll show codons, how exactly the connection between DNA, RNA to amino acids connectivity. And then we'll begin amino acid properties because that's much more complex. Okay.
Sir, I have a question that in the DNA structure you have double helix structure DNA, and there is DNA experiencing hydrogen bonding. So there is a energy profit in DNA structure. But why do you not expect a double stranded structure like DNA in RNAs? Why RNAs all time a single stranded? They can form also double stranded structure.
No,
they can
but
experience.
see, why the cell has to see this is only messenger, right? So you have to copy just one strand. Okay, you don't have to copy this complementary strand. So then you won't have the complementary strand synthesized. Okay, of course, if we have complementary strand, then obviously it will form nice double-stranded RNA. But you won't have complementary strand at all. Okay, RNAs don't have complementary strands. Only thing is that some viruses, you have RNA viruses. So where actually you can synthesize copy of RNA from another RNA. So that's only place where we can really think of. Otherwise, our cells do not have a complementary RNA strand for every messenger RNA because you don't require it all. Okay, that's why I'm telling you the problem even with DNA, but we never really study that because DNA automatically comes with a complementary strand. So RNA doesn't have come. Okay. So in the same context, I think we should read what's called palindromes. Okay. Palindrome sequences are actually kind of self-complementary. Okay, so example, let's say A, T, G, C, C. Okay, so then this strand will have G, T, G, C, A, G. Okay, so if you see, this DNA sequence, okay, so A, T, G, C, A, C. So the second half is exactly complementary to the first. Okay.
Yes,
Sorry, you're right. You're right. Sorry, you're right. So this is self-complementary. So it doesn't have actually a complementary strand. Okay. So it can actually, by itself, is a complementary strand. Okay, do you see that the if you have to write the complementary strand, so you write A, T, G, C, A, C, G, T, G, C. You see the same strand I have written bottom means that you synthesize just one strand, put it in test tube. It formed double-stranded DNA.
yes,
okay. So unlike a random sequence, let's say Okay, let's say if I have let's say poly, just for the fun. So here, if you have poly A and test tube, you need poly T, then only forms double stranded. But here, interestingly, this strand is exactly same as this. So one strand becomes common. So this is a palindromic sequence. Okay, so sometimes you have fun with words, right? Kind of words. It's a palindrome.
Yes, sir. Yes, sir.
So like that, so these are palindromic sequences. So, and also there is something else cruciform. Let's read the terms. Cruciform. Okay, so these are interesting structures one needs to. Okay, I think that's all for today. So next class we will
Sir, I have a question.
brief introduction with the central document. Yes.
Sir, sir, I want to say that the uracil is came from only cytosine, prepared from cytosine and thymine, or it is prepared on another way in cell? Because there is only methyl difference. So is it prepared from thymine, or there is a whole lot of different mechanism for preparation of uracil from the DNA?
I don't know. So individual bases needs to be synthesized, right? Okay. So I'm not sure how exactly synthesis. Most probably, maybe yeah, chemical modification of thymine could be one of the routes. I'm not really sure. Because every base you need to synthesize, or every nucleotide inside the cell. So most probably, yeah, maybe methylation of thymine, etc. Okay, or it could be somewhere. Sometimes it's the opposite. Maybe uracil is easy to synthesize, right? Because it has one methyl group less, right? You need to add methyl group. So maybe that's maybe thymines are synthesized from uracil. I would think like this, rather than you synthesize with methyl and then remove methyl. It's more energetic. So maybe uracil is actually synthesized first, and then you add thymine and then use that for DNA. Most probably. Okay.
Okay.
It seems like cytosine.
Yeah. Cytosine to.
If cytosine is got hydrolyzed, then it can convert it to uracil.
Yeah, but cytosine to thymine, you have again carbonyl group you need to replace. That NH2 we have to remove and then carbonyl. But uracil and thymine, you have only this methylation. Yeah, but it could be, yeah, so these are biosynthetic pathways. One needs to really see that how one metabolites. So these are, we call metabolites, okay, are molecules which are synthesized. So, but you need to really find out. I'm not really sure that how exactly that.
Sir, in the previous classes, you said that all the cells are having the same DNA, but the way they are expressed themselves is different. So we can we say that all the cell are having different messenger RNA, because that is the reason, that's why they are expressing
But
in a different way.
that's what I meant. So different genes are expressed means that different messenger RNAs.
Okay.
Yeah. So because every cell has to synthesize its own messenger. So you're absolutely right. I was also right. That DNA is the same, but which genes expressed means that each gene 1 has a messenger RNA 1, gene 2 has messenger RNA 2. So which genes are expressed, the corresponding messenger RNAs are there. These are, mind you, these are time dependent. This is called spatio-temporal. Okay. So maybe cell depending on what state you are in. Okay. What state the cell is in. Maybe it needs to have messenger RNA only for short time. Okay. So when it is required, it synthesizes. But once its job is done, so you have to actually recycle it. Okay. Otherwise, it will be floating forever. So you have to consume this. You have to degrade this messenger RNA so that the protein is not made. Otherwise, ribosomes will constantly synthesize. So this is something called feedback and whatnot. We will discuss as we go deeper into the course. Okay, I think that's all for today. Thank you. So next class will be on
Thank you, sir.
Monday. Okay. and happy puja to everyone, especially in college.
Same to you, sir.
I'm sure everyone is celebrating at home. People in the college celebrating the house. Okay. Okay. Thank you.
Thank you, sir.
Okay.
Thank you, sir.
Bye, sir.
Thank you.